Hi, my name is Janae Heiss, and this video is on the topic of DNA replication and PCR. I am doing this video for my master's degree in Applied Ecology and Conservation Biology at Frostburg State University. My thesis for the degree is on cannabis cultivar genotype and chemotype, which is basically how different types of cannabis look in their genetic composition and their chemical composition. This concept, DNA replication and PCR, pertains to my thesis because knowing how DNA replicates in a cell is a good contrast to knowing how DNA replicates in PCR. My thesis actually involves PCR and using it to amplify specific microsatellites in order to determine the size or sizes of the microsatellite alleles present in each sample. This video, per Dr. Putoff's request, is 60 minutes long and does not use slides or notes. DNA replication and PCR. So let's start with DNA replication and all of that concept. First, we're going to look at the molecular structure of the chromosome and the molecular structure of DNA. This gets into the mechanism of how we get phenotypes and how those genes are inherited. One of the first things to look at when studying DNA is to look at the history of it. So, how did people figure out that DNA was that genetic material? Gregor Mendel, like we talked about in Mendelian genetics, was the first one to figure out alleles. He determined that there were two main forms of a particular trait, and that those traits were inherited through genes. Additionally, he determined that both parents contribute one of these alleles to their offspring. So the mom contributes one allele, and the dad contributes one allele. After Gregor Mendel, we have Thomas Hunt Morgan. He figured out that chromosomes were the thing that were inherited by the offspring. Thomas Hunt Morgan was a scientist in the 1900s who did work on chromosomes, sex-linked traits, genetic linkage, and he hypothesized crossing over. He determined that chromosomes were the thing that were inherited so that you get one chromosome from your mom and one chromosome from your dad. So now that we know that chromosomes exist, the next step in figuring out the genetic puzzle was to determine what a chromosome was made of. They found that chromosomes were made of protein and DNA. That sparked off the next debate, and that was trying to figure out whether protein or DNA was the genetic material. Oswald Avery and others in the 1930s had experiments that demonstrated that DNA was the thing that was taken up in the transformation of bacteria. Transformation is how bacteria take up DNA from their surroundings, genetic material, and they use it to express new traits. These 1930s experiments determined that DNA was the thing that was taken up. However, it wasn't until 1952, with Alfred Hershey and Martha Chase, that they determined that the thing that was transforming that bacteria was definitely DNA. They verified that the genetic material was DNA from experiments on bacteriophages. Bacteriophages are viruses that infect bacteria. In this, the phage replicates itself in the bacteria and then causes that cell to burst. They determined that the thing that was replicating inside the bacteria was genetic material, and that was transferred to the next generation of bacteriophages. These and other experiments showed that DNA was the genetic material. DNA had the instructions that gave rise to all the products that make cells be able to function. In the history of genetics, chromosomes were shown to be made of proteins and DNA. In the experiments that I talked about earlier, they showed that DNA was the main molecule for genetic inheritance. So now that they figured out that DNA was that molecule for genetic inheritance, the next step was to figure out what exactly was it made of. So they needed to figure out the molecular structure of the DNA. This leads us to the history and debate between Maurice Wilkins and Rosalind Franklin, and James Watson and Francis Crick. This is a classic story in the history of science. So there were 4 main scientists in this story. There was Maurice Wilkins, who was a scientist in a large lab that used a technique called X-ray crystallography, and there was Rosalind Franklin in his group. So X-ray crystallography is a technique that's used to determine the structure of a molecule by shining X-rays at it. This molecule was put in the form of a crystal, and then X-rays were shined at it, and it bounced off onto a sheet of X-ray film that was put behind the crystal. Based on the patterns in the X-ray film, the scientists could determine the structure of the molecule. This technique was mainly used to determine the structure of proteins. But Rosalind Franklin wanted to use X-ray crystallography to study DNA. She was the first person to ever take a photo of the structure of DNA, and she made the famous photo called Photo 51. This photo showed patterns that indicated that DNA was a helix shape, and it was likely a double helix, so 2 helixes. And then the phosphate groups in the DNA were found on the outside of the molecule, not the inside. Before all of this, before her work, people figured out that there were phosphate groups, and they knew that DNA was negatively charged, but they weren't sure of how all the stuff worked out. They weren't sure how the pieces fit together. So this leads to her attempts to figure it out. Rosalind Franklin was working on it, and so were Watson and Crick. So Rosalind Franklin submitted her progress report to an individual in the Medical Research Council, and that individual shared her data without her permission to both Watson and Crick. Watson and Crick found out about Photo 51 from this individual, and they used the information to piece together all the parts of DNA. They figured out the structure where all the pieces fit together. And then they published the structure into one of the 2 most prestigious journals in biology at the time, Nature. They did this in 1953, and after which, Rosalind Franklin wrote an article where she told them how they got the structure wrong. And she was right. Later, after they determined the structure of DNA, in 1962, Watson, Crick, and Wilkins were all awarded the Nobel Prize in Medicine. Rosalind Franklin had passed away by then. Otherwise, she would have been included. Unfortunately, she died of ovarian cancer before the Nobel Prize could be awarded. In scientific textbooks, Watson and Crick are typically known as the people who discovered the structure of DNA. Next, let's get into chromosome structure. So there are 2 main types of chromosome structure. Bacterial chromosomes are smaller than eukaryotic chromosomes. They have a circular ring shape, and they don't get packed tightly like eukaryotic chromosomes do. On the other hand, eukaryotic chromosomes are very large. They have multiple chromosomes that are all packed into a small space, the nucleus. There's lots of DNA in one chromosome, and most cells have dozens of chromosomes. There's so much DNA that it's surprising it can even fit into the nucleus. So, to actually get it to fit into the nucleus, the cell uses a complex, multi-layered packaging system that organizes the DNA. So, the first step for packaging the DNA is the pure DNA molecule. It's a double helix that's about 2 nanometers wide. And then, to package it, we need to wrap that double helix strand around a histone core. Histone proteins are globular proteins with long tails. 8 of them come together in a bead-shaped histone core, and the DNA wraps around that core 2 times. The long strand of DNA wraps around a bunch of histones. Once all the DNA is wrapped around the histones, the DNA gets shorter. And the complex of DNA wrapped around histone proteins is called a nucleosome. So that's how much detail Bio 149 covers. To get into more detail on that, let's look at histone proteins in detail. Histone proteins are designated histones H1 through H5 for the 5 families of histone proteins. Histones H2A, H2B, H3, and H4 are the core histone proteins. H1 and H5 act as linker proteins. The nucleosome core is made from 2 dimers of H2A and H2B, and from an H3-H4 tetramer. The histone proteins are all positively charged, so they wrap around the negatively charged DNA like magnets. And like those magnetic charges, they keep it in place. The H1 linker protein locks that DNA strand onto the histone, keeping it in place. To put together the histone, 2 H3 and 2 H4 proteins come together to make a tetramer. This tetramer is roughly a half-circle shape. And then that tetramer goes onto the DNA strand. Then H2A and H2B come together and make a dimer. The other set of H2A and H2B come together to make a second dimer. Those 2 dimers come together on the other side of that histone core, and then it wraps and makes a circular protein that wraps the DNA. And lastly, the H1 protein locks that DNA into place. A histone core can wrap about 150 base pairs of DNA. A bunch of nucleosomes are wrapped around the DNA strand. So across the DNA strand, it's all wrapped in nucleosomes. So there's complexes of histone proteins. And between each nucleosome, there's about 50 base pairs that separate each. So these 50 base pairs are called linker DNA. Histone proteins can be methylated or acetylated. To methylate histone proteins, it tightens the winding of the histones, so that way the histones can't be unwound as easily. Acetylation, on the other hand, weakens the attraction between the histones and the DNA, so that way the cell can unwind that DNA and get access to it much more easily. Acetylation happens when cells need that DNA for gene expression. So we've got the DNA condensed in histones. So once we've got the DNA wrapped around the histones, it's called a nucleosome. And nucleosomes are 10 nanometer fibers. And the structure of nucleosomes is called euchromatin. After we have the nucleosomes, then we coil them around each other. And this creates a 30 nanometer fiber. This 30 nanometer fiber is called heterochromatin. Both the 10 nanometer state and the 30 nanometer state are called chromatin. 10 nanometer is euchromatin, 30 nanometer is heterochromatin. When the cell is in interphase, the DNA is just kept in that chromatin state. It's loosely wrapped and easily accessible. But when the cell gets ready to divide, it needs to wrap its DNA even tighter. It condenses into the chromosome shapes that are classic to genetics. So it's the X shape. So more proteins called scaffolding proteins have to be added, and the fiber loops around the scaffolding proteins in a daisy chain pattern. So that 30 nanometer fiber just loops around the scaffolding proteins. And this makes a 300 nanometer fiber. And this fiber coils up even more, and it produces the chromosome that's classic to mitosis and meiosis. Examples of scaffolding proteins include things like condensin, topoisomerase, and KIF4. None of these proteins are typically mentioned in a Bio 149 class. So, to summarize all the levels of DNA condensing, we have the DNA fiber, which is 2 nanometers. Then we scale up as histone proteins wrap around it and make a nucleosome. Those nucleosomes wrap around each other to make heterochromatin. And then, if we're going to mitosis, those DNA coils into a daisy chain pattern. So, around the scaffolding proteins. And this makes an even thicker fiber. And that can even coil up even more to make the chromosome. So now that we've gone through the chromosome structure and how DNA gets packaged up to make the chromosome, next let's get into the structure of DNA. So what is DNA made of? DNA is a double-stranded molecule. It's in the shape of a twisted ladder or a double-stranded helix. These strands go in opposite directions from each other. So we say that the strands are anti-parallel. The ends of the strands are called the 3' end and the 5' end. One strand runs from the 3' end to the 5' end, and the other strand runs from the 5' end to the 3' end. So for example, with this molecule, this might be the 3' end and this might be the 5' end. And then over here we'd have the 3' end of that one and the 5' end of that one. So the phosphate groups run on the outside of the molecule. Because the phosphate group is a phosphate surrounded by negatively charged oxygens, there's a bunch of negative charges running down the sides of the DNA molecule. DNA is lined with these phosphate groups on either side of the molecule. DNA is a double helix with a sugar phosphate backbone. So each of these phosphate groups, which are represented as circles here, is attached to a 5-carbon sugar. The 5-carbon sugar in DNA is called deoxyribose. And this 5-carbon sugar is connected to a nucleobase. There are 4 main different bases: adenine, cytosine, guanine, and thymine. These bases can be categorized into being purines or pyrimidines. Adenine and guanine are purines. Cytosine and thymine are pyrimidines. The difference between the two of them are in the number of ring structures. But to have a little verbal thing for the differences, pyrimidines have a Y, and so do cytosine and thymine. So you can remember the difference between the two of them and that they all have a Y. Purines have 2 different rings, so they're bigger. But pyrimidines only have 1 ring. So adenine and guanine have 2 rings, and thymine and cytosine have 1 ring. The bases are bound to each other by hydrogen bonds. So when we're talking about the structure of DNA, and we have these bases, they are connected to each other by hydrogen bonds. Hydrogen bonds aren't permanent. They're weak on their own. But because there are so many hydrogen bonds in DNA, it keeps those strands associated with each other, so it keeps them stuck together. All the nucleotides on a strand are held together by phosphodiester bonds. So those bonds between the phosphate group and the deoxyribose sugar are phosphodiester bonds. When we're talking about nucleobases, A always binds with T, C always binds with G. There are 2 hydrogen bonds in the A and T pair, and there are 3 hydrogen bonds in the C and G pair. So whenever we have those binding, they bind to each other because of the number of hydrogen bonds that are there. So a T couldn't bind with a G as easily because the T only has 2 hydrogen bonding sites, while the guanine, the G, has 3. So AT bonds are easier to pull apart than CG bonds because they have fewer hydrogen bonds. And this comes into play when we're trying to pull apart the DNA. So it also comes into play when we're trying to do things like calculate melt temperatures for primers and PCR, which is going to come into play toward the end of this video. Not calculating the melt temperatures, but just melt temperatures, the concept. So we've got the DNA structure. Now we need to actually replicate it. So let's ask the question, why exactly do we need to replicate DNA? And the answer to that is, cells die in your body every day, and new cells are needed to replace them. To replace cells, your body needs to go through mitosis. And to do mitosis, you need duplicated DNA or replicated DNA. So, to be able to even get new cells, we need to have replicated DNA. In mitosis, for the cell to divide, it needs to go through synthesis phase, or S phase. In this phase, all the chromosomes in the cell are replicated. And replication is the process through which DNA is copied. So, DNA replication, DNA copying. We make 1 DNA molecule into 2 DNA molecules. It's important that that DNA be copied perfectly and accurately. We don't want mistakes. Mistakes made when DNA is replicated are passed from that parent cell to the daughter cells, and those daughter cells pass it on to their daughter cells. So that's bad. Those mutations could lead to things like cell death or cancer. The double-stranded structure of DNA allows those strands to be pulled apart so that the cell can automatically build a new copy from that parent strand. So when we get that DNA, it's two-stranded and we pull it apart. And then we copy the parent strand to get 2 new daughter strands. The new daughter strands are composed of 1 strand from the original parent and 1 strand that was made in DNA replication. To do DNA replication, you need 3 main things. You need a template strand of DNA. This is also called the parent strand. It gives you something to copy. So if we were to do a printer analogy here, the template strand of DNA would be the paper that you wanted to copy. DNA polymerase is an enzyme that builds DNA. It's the second thing you need. It acts like molecular machinery to do the copying. So DNA polymerase would be the copier in that copying analogy. And then the last thing we need are nucleotides. Nucleotides provide the building blocks for that DNA strand to be built. They're the A's, T's, C's, and G's that you need to make new DNA. And they'd be the last part of that printer analogy. They'd be ink. So to make a new DNA strand, we need a template strand, DNA polymerase, and nucleotides. So let's get into the concept of DNA polymerase and DNA replication. DNA polymerase occurs in a big complex of enzymes. It's a molecular machine that adds nucleotides to the new strand of DNA. But there's a catch. DNA polymerase is only able to add nucleotides to the 3' end of the strand. And this 3' end of the strand is this side with the OH group at the end. Because we have 2 strands that need copying, we need to pull those strands apart. And these strands being pulled apart form something called a replication bubble. The origin of replication is where those strands pull apart. This is where DNA polymerase sits and starts building new DNA. So, like I said earlier, DNA polymerase only adds nucleotides on one end. So on that one end, it gets built really easily. But the other end is kind of backwards. This leads to one strand being built faster than the other strand. The faster strand is called the leading strand, and the slower strand is called the lagging strand. The leading strand is just made in one attempt. It's done. But for the lagging strand, it needs to be built in sections. So it just hops on and hops off the DNA strand, and it's built section by section. These sections are joined together by other enzymes. Most DNA polymerases, including this one, requires a primer and a DNA template to set down to start elongating DNA. Primers are little bits of RNA that tell DNA polymerase where to begin. To copy DNA, DNA polymerase needs a spot to start, and the spot where DNA polymerase starts is called a primer. Primers are little bits of nucleic acid that form the starting point for DNA polymerase to set down. And because these primers are made of RNA, we don't have the same bases. So RNA has similar bases to DNA, but instead of thymine, it has uracil. So when we get the primer on and it's added, it gives polymerase a spot for DNA to just set down and go. The leading strand is good, but then the lagging strand has to be made a little differently. So instead of just the one primer, we get a bunch of different primers made. And each of these primers has a spot, is like the spot where DNA polymerase can set down and build another section of DNA. And eventually, because it gets to those spots and it copies them, we copy the whole chromosome. Okazaki fragments are the name of the fragments that are made on the lagging strand. These fragments are named for the husband and wife team from Japan that found them, Okazaki. So the rate of elongation, once we get the DNA replicated, DNA polymerase to set down and start replicating, is 500 nucleotides per second for bacteria and 50 nucleotides per second for humans. So we can copy DNA pretty fast. And one other thing that's really important with DNA is that it's universal across all organisms. So we have the same DNA in us as bacteria have, as mammals have, as reptiles have, as anything has. And it uses about the same form of instructions. So we can use it for a lot of different ways. We've got a couple things that are needed for DNA replication, aside from just DNA polymerase. We've got helicase. The enzyme that pulls DNA apart is called helicase. It unwinds the 2 strands and separates them out. And it spins really quickly to unwind them. There are also proteins called single-stranded binding proteins that bind to those strands of DNA and keep them apart. And then there are also other proteins that are important. So let's just summarize. We've got DNA polymerase. It's the main enzyme for DNA replication. We get the leading strand, which is made in one shot, and the lagging strand that's built with a bunch of different primers backwards because it needs to go in the 3' direction. So this primers need to be taken off eventually, and those primers are replaced with DNA by a different enzyme. And at the end of DNA replication, there are 2 copies of DNA. So that was DNA replication at the Bio 149 level. All of the topics and concepts there were at that level and were covered in Bio 149 classes. So let's get into more detail. This version of DNA replication that I'm going to go over next will be focused on eukaryotes because my thesis is on a eukaryotic organism. So, starting with DNA polymerase. It's not just the one molecule. There's more than one kind of DNA polymerase, and all the DNA polymerases are composed of a complex of different enzymes that carry out DNA replication. Most types of DNA polymerase can't synthesize new strands by themselves, and they are only able to extend an existing DNA strand. So we covered that part in the Bio 149 portion. And to start replication, they need that short fragment of RNA, the primer, that gets paired with the template DNA strand. DNA polymerase replicates DNA by extending the 3' end of the primer, adding new nucleotides matched to the template strand, making phosphodiester bonds between each new nucleotide. The energy needed for making new nucleotides and joining them onto that DNA comes from hydrolyzing the excess phosphate bonds on the unincorporated nucleotides. DNA polymerases are really accurate enzymes. Some of them even have proofreading abilities, which allows them to remove nucleotides from the end of the strand and fix mismatched bases. Post-replication mismatch repair mechanisms monitor this DNA for errors, and these mechanisms are capable of distinguishing mismatches in the newly synthesized DNA strand and telling it apart from the original strand sequence. Basically, they allow the DNA polymerase to be accurate to the degree of about 1 mistake for every 100 nucleotides that are added to the DNA molecule. When we're talking about DNA replication, there are 3 main steps: initiation, elongation, and termination. Step 1 of DNA replication is initiation. The cell needs to replicate its DNA so that it can divide. Once replication is started, it needs to go all the way to the end of the process. So it's an all or nothing. The first part of initiation is forming the pre-replication complex in early G1. So the pre-replication complex is a large complex of initiator proteins that are put into the DNA at different locations. Those locations are called origins. So they make the origin of replication. The origins that those initiator proteins use are AT-rich. That means they have a lot of adenine-thymine bonds. Adenine and thymine base pairs have 2 hydrogen bonds instead of the 3 hydrogen bonds that are in cytosine and guanine base pairs. So those are easier to pull apart. The AT bonds are much easier to separate, so they're easier to be used for the origins of replication. In eukaryotes, the origin recognition complex assembles these initiator proteins into the pre-replication complex. This pre-replication complex binds onto the DNA. The proteins called CDC6 and CDT1 associate with the bound origin recognition complex and make a bigger complex. And this complex loads an MCM helicase protein onto the DNA. This complex makes the origin of replication. Helicase unravels the DNA helix, and once helicase is on the DNA, it's the ending of the pre-replication complex formation. So we've got that helicase there, it's not started yet, but once it is started, it'll unwind that DNA. And it makes that whole pre-replication formation complex complete. After the pre-replication complex is formed, the complex is activated by cyclin-dependent kinases. This activation makes the expression of genes that code for components of the DNA synthesis machinery. So we get more replication factors that unwind the DNA and add more DNA polymerases to the DNA. And this leads to the initiation of DNA synthesis by making an active replication fork. This is on the initiation complex. So earlier we made the pre-replication complex and we had cyclin-dependent kinase activity. These cyclin-dependent kinases, they activate and they cause the assembly of the initiation complex. This initiation complex is formed at the origin of replication, and it's made up of approximately 100 proteins. Once we get the initiation complex formed, it displaces the CDC6 protein and the CDT1 protein from the origin replication complex. So this inactivates and disassembles the pre-replication complex. So we get rid of the pre-replication complex and make up the initiation complex. CDC45 protein is an important one for the initiation complex. It assembles at the origin of the chromatin, and it's required to start replication. CDC45 is needed to load other proteins onto the chromatin, including DNA polymerases. It combines with other proteins like MCM helicase to form CMG helicase. Once we get the initiation complex loaded onto the origin, it activates helicase, and helicase causes the DNA double helix to unwind. This initiation complex loads primase and DNA polymerase onto the DNA, and the pieces of the initiation complex remain associated with the replication forks as they move out from the origin of replication. Once we get this initiation complex formed, the cells move to S phase. Step 2, that's elongation. So we've got that initiation complex, and we start elongating the DNA. Once the cells pass to S phase, the initiation complex becomes called a replisome. This replisome coordinates DNA replication on the leading and the lagging strand. DNA polymerase has 5' to 3' activity. It requires a free 3' hydroxyl group before synthesis can be initiated. The new strand is synthesized in the 5' to 3' direction. Because nucleotides can only be added onto the 3' end of the DNA strand, this is often confused. So DNA is read by DNA polymerase in the 3' to 5' direction, and the new strand is synthesized in the 5' to 3' direction. Since the leading and the lagging strand templates are oriented in opposite directions at the replication fork, the big issue now is to figure out how to get synthesis of the lagging strand DNA, whose synthesis of DNA is opposite to the direction of the replication fork. All living things use a primase to synthesize a short RNA primer. This RNA primer has a free 3' OH group, and this primer is what's used to start DNA synthesis. Primer template junctions allow DNA synthesis to start, and the DNA polymerases elongate this RNA primer with DNA. Once the two DNA strands are separated, primase adds that RNA primer to the template strands. The leading strand gets one RNA primer, and the lagging strand gets several RNA primers. The leading strand is continuously extended from the primer, and the lagging strand is extended in short, separated sections from each primer. Completed replicated sections on this lagging strand are called Okazaki fragments. DNA polymerases are really important to replication. So after DNA primase has created the RNA primer, DNA polymerase alpha elongates the primer for about 20 nucleotides. Then DNA polymerase delta takes over because it's a faster DNA polymerase. It allows faster replication. DNA polymerase delta interacts with the clamp protein pCNA, and it gives it increased processing ability. It's thought that DNA polymerase delta replicates the lagging strand, while DNA polymerase epsilon replicates the leading strand, but this is still being debated. What we do know is that there are three main types of DNA polymerases that are involved in replication: DNA polymerase alpha, DNA polymerase delta, and DNA polymerase epsilon. As DNA synthesis continues, the original DNA strands continue to unwind on each side of the replication bubble. This forms a replication fork with two prongs. Eukaryotes have long linear or chromosomes, so they initiate replication at multiple origin spots. Step 3 is termination. This is where we end DNA replication. Eukaryotes initiate DNA replication at multiple points in the chromosome. So replication forks meet and they terminate at many points in this chromosome. And because eukaryotes have linear chromosomes, DNA replication can't reach every point in the end of the chromosomes. So there's still the very edges of the chromosome that haven't been replicated. And due to this problem, DNA gets lost in each replication cycle from the edges of the chromosome. To fix the problem, we have telomeres. Telomeres are regions of repetitive DNA that are close to the ends of the chromosomes, and they help to prevent loss of genes through the shortening of the telomeres. So, shortening of the telomeres is a normal process in somatic cells. But as a result, those cells can only divide a certain number of times before DNA loss stops them from dividing any further. This loss is known as the Hayflick Limit. It's that stopping point where they can't divide any further. Telomerase extends the repetitive sequences of the telomere region so that it stops this degradation. So once we have telomerase that's taking care of the edges, we get the main termination. Termination requires two things. It requires that the progress of the DNA replication fork has to stop, or it has to be blocked by something. So termination at a specific point involves the interaction between a termination site sequence in the DNA and a protein that binds to the sequence to physically stop it from replicating further. This usually happens when DNA reaches the next RNA primer. So once it reaches that last DNA primer, it pops off, and then we have the next thing. RNase removes those primer RNA fragments, and then another DNA polymerase comes in and it fills the gaps left by those primers. This DNA polymerase is able to fill the gaps, but it can't do it completely. It leaves a nick between the one edge of the sequence and the next. So we have one nick on the leading strand because there's only the one RNA primer that's needed, and we end up with several nicks on the lagging strand. This is where ligase comes in. Ligase works and it fills all of these nicks in, making this a complete, newly replicated DNA molecule. And that's how organisms replicate their DNA. So now we've made it through DNA replication. We've got through all of the steps that are needed to make a new DNA molecule in the cell. So now we're on to PCR. PCR is how we make new DNA in a lab. Before we get started on going through PCR, I would like to point out that Bio 149 classes do not typically cover PCR. There's no lecture portion that covers PCR as far as I know, and there was a lab portion that glossed over the concept, but it has not been taught since the fall of 2019. And at that point in time, I was teaching it. PCR is a lab technique that's used to amplify a specific target region of DNA. By amplify, we mean make copies. So PCR makes millions or billions of a specific DNA spot, a region that the researcher is interested in. PCR can be used for things like determining gene functions and matching crime scene DNA with suspects. The goal of doing a PCR is to make enough of the target DNA to either analyze it or use it in some other way. DNA amplified by PCR can be sent for sequencing, visualized by gel electrophoresis, or cloned into a plasmid. So let's get into the history of PCR. PCR was invented in 1983 by a man named Kary Mullis. He was an American who was a biochemist at the Cetus Corporation. He was trying to increase the demand for oligonucleotides for his lab. Oligonucleotides are short pieces of DNA and RNA. Because DNA is very specific, an oligonucleotide that's maybe 20 bases long is still enough to specify some spot on the DNA. He determined that if he could get oligos that were specific to and framed certain spots in his DNA, then he could get that spot in the DNA to be copied. He came up with a mechanism for this, and he wrote it down in his cabin. Thanks to his efforts, he was awarded the Nobel Prize in Chemistry in 1993. Like DNA replication, PCR requires three main things to make more DNA. It needs a template strand of DNA. So while in DNA replication, you'd have the organism's DNA, PCR, you'd need some sort of DNA sample. So you'd need to extract some DNA. You also need nucleotides. Both DNA replication and PCR need nucleotides. And then the last main thing is polymerase. You need some sort of polymerase for PCR. While in DNA replication, you need DNA polymerase, PCR uses other types of polymerases. The most common type of polymerase used in PCR is Tac Polymerase, but my research uses Tac Polymerase. Because there's just the two polymerases, I'm going to go over both of them later in this video. And unlike DNA replication, PCR needs one more thing. It needs primers. While in DNA replication, primase can make primers, PCR requires that there are specific primers for the target sequence that are already made. So let's just make sure we know about some stuff. So the template DNA, it needs to be extracted from an organism. The organism could be anything, but there just needs to be DNA that has a sample taken, and then that DNA sample gets purified. And then the nucleotides are the A's, T's, C's, and G's. They get built up into a new strand of DNA. In addition to needing the DNA template and nucleotides for PCR, we also need polymerases. So like DNA replication in an organism, PCR requires a DNA polymerase enzyme that makes new strands of DNA and uses the existing strands as a template. So the first one we're going to go over is Tac Polymerase. The DNA polymerase that's the most commonly used in PCR is called Tac Polymerase. This is after the heat-tolerant bacteria from which it was isolated, from Thermus aquaticus. It takes the T from the thermus and then aquaticus, AQ. Thermus aquaticus lives in hot springs and hydrothermal vents. Its DNA polymerase is very heat stable and it's most active around 70 degrees Celsius. Its heat stability makes Tac polymerase ideal for PCR. There's also Top polymerase. Top polymerase is from Thermus thermophilus, strain HB27. Thermus thermophilus was originally found in a thermal vent at the bottom of the seafloor in Japan. Top DNA polymerase is similar to other DNA polymerases from Thermus species. It's about 87% similar to Tac DNA polymerase. This DNA polymerase is most active at about 76 degrees Celsius. So it is actually is heat stable at a higher temperature than Tac polymerase. Because high temperature is used in PCR to denature the template DNA, whatever polymerase that you use for PCR needs to be able to withstand temperature changes. In addition to polymerase, you also need PCR primers. Polymerase can only make DNA if it's given a primer. That primer is a short sequence of single-stranded DNA about 20 base pairs long. It's an oligonucleotide that provides the starting point for DNA synthesis. In a PCR reaction, the researcher needs to carefully design the region of DNA that will be used to make up the primer, and then pick out the region of DNA that will be amplified by that primer. So, the two primers that are used in each PCR reaction are designed so that they flank the target sequence. In other words, they frame that target sequence so you only get the sequence that you want copied. You get one on either side of the DNA, and they bind to the template by complementary base pairs. Now that we have the things that we need to do PCR, let's look at the steps that it takes to do PCR. So we've got the key ingredients. We have the polymerase, the primers, the template DNA, and the nucleotides. And then we put those into a tube. Those pieces, along with cofactors that are needed by the enzyme, are put through repeated cycles of heating and cooling that allow the DNA to be synthesized. The basic steps of a PCR reaction are denaturation, annealing, and extension. In denaturation, the step is around 90 degrees to 96 degrees Celsius. In this step, the reaction is heated to separate or denature the DNA strands. So we heat up the DNA and we separate the strands. This makes the DNA single-stranded, and it allows it to act as a template for the next step. Denaturation takes about 20 to 30 seconds depending on the reaction. Annealing takes place at around 55 to 65 degrees Celsius, and this varies a lot depending on what primers you have. In this step, the reaction is cooled down so that the primers can bind to their complementary sequences on the target DNA. It takes about 20 to 40 seconds, and the temperatures that are used in this step are specific to the melt temperatures of the primer selected. So when you make up a primer, the specific primer combinations have a specific melt temperature, and this is what you use for the annealing temperature. So it's based on the annealing temperature. You base the annealing temperature on it. For extension, the third step, the temperature is raised to about 72 degrees Celsius. This allows the polymerase to extend the primers out and make the DNA. Once the primers are fully extended, the new strands of DNA are synthesized. It takes about 30 seconds for every 500 base pairs of product that are needed. And it has the most variation time. So extension, if you want to make a small product, you only need to go for a little bit of time. But if you want to make a large product, you need to increase that time to whatever you need. So this 3-step cycle repeats 25 to 40 times in a typical PCR reaction. And it typically takes about 2 to 4 hours, depending on the length of the DNA region being copied. This target region can go from being a few copies to billions of copies. So it greatly amplifies the amount of DNA present in a sample. In addition to these 3 main steps, there are some other steps that are needed for PCR. Some polymerases require an initialization, where the initial temperature of the reaction is held at the denaturation temperature for an extended period of about 1 to 10 minutes before the reaction starts. So my research has this initial step, and it's used to prime the polymerases for the reaction. A final elongation can be used to make sure that the DNA is fully elongated. And then lastly, there's the final hold step. This final hold step allows for some short-term storage of the PCR products after the reaction has completed. So once we've reached final hold, we can just keep it there to store the PCR products at about 4 degrees Celsius and keep them stable until we can get them later. PCR can make billions of copies because it's not just the original DNA that's used as a template each time. Instead, the DNA that's used from the last cycle can be used as a template for the next cycle. So each time you do a cycle, you increase the number of templates that are available to be duplicated. So there are a bunch of primer copies, polymerase enzymes, and nucleotides floating around in the reaction. So the number of DNA molecules can double in each round of cycling. After we've got the PCR complete, we need to get it and view the results somehow. So we can visualize it using a technique called gel electrophoresis. Gel electrophoresis uses electricity to take and pull DNA fragments through a gel matrix. The gel electrophoresis separates the DNA according to its size. A standard DNA ladder is typically included on the gel so that you can see the size of the fragments in the PCR sample. DNA fragments of the same length in a sample form a band on the gel, and that DNA band contains a lot of different copies of the target DNA region. So the amount of copies is a big part of why PCR is an important tool. PCR produces enough copies of a DNA sequence that we can visualize it and manipulate that region of DNA if we want to. PCR produces enough DNA to be used in other techniques too. It can be visualized with gel electrophoresis, and then it can be sent for sequencing, or it can be digested with restriction enzymes and cloned into a plasmid. And because of its amazing variety of uses, PCR is used in many research labs. It has practical applications in forensics and genetic testing. In my research, PCR is being used to determine the genetic diversity in hemp samples. One big use of PCR is in forensics. After DNA is found in a crime scene, technicians do an analysis of the DNA sample like I talked about earlier. But instead of using the one DNA marker, they use many different markers, and they compare the DNA samples between the crime scene DNA and the suspect's DNA. The markers used in a typical forensics analysis don't come in two different forms, like Mendelian genetic states. Instead, they have a lot of different alleles that vary in little increments. They are most commonly in the form of short tandem repeats, also known as simple sequence repeats or microsatellites. Microsatellites consist of many repeating copies of the same short nucleotide sequence. They are typically 2 to 5 nucleotides long, and one microsatellite allele could have like 15 repeats, another could have 12 repeats. There are a lot of different repeats on that same sequence, and the number of repeats can determine the size of that final product. After we examine the differences between the samples for a lot of different markers, the researchers can build unique genetic fingerprints from those DNA samples. In a typical human forensics analysis that uses 13 markers, the odds of two people having the same DNA allele combinations are less than 1 in 10 billion people. Forensics analyses are used to look at crime scenes, establish paternity, and to identify human remains after disaster scenes. One lesser known use of DNA forensics is for drug testing, so they can be used to categorize cannabis into drug type and fiber type. Researchers use different microsatellites to build profiles of the seized hemp samples and determine whether those cannabis samples are illegal or not. So to summarize PCR is short for polymerase chain reaction. It's a technique that's used to make many, many copies of a specific DNA region in a laboratory setting. PCR needs a specific DNA polymerase that can withstand high temperatures, and it requires specifically designed DNA primers that are for a DNA region that you want to copy. In order to do PCR, the reaction is repeatedly cycled through a series of temperature changes, and it's used to break apart the DNA strands, anneal the primers onto those DNA strands, and elongate those strands. And once we've got those, we just cycle it again and again and again. And in these cycles, many copies of the target region are produced. Once we have those copies, and we have all of that DNA, we can use it to do other things like identification and genetic testing. Since I'm still here, let's just do the finishing touches. Thank you for watching this video. My name is Dna Heiss. I hope you learned something about DNA replication and PCR while watching this. This is the last video in the series, so guess I won't see you later. No, bad.
Good job of applied, dude. Oh my gosh. Guess I won't see you later. You're going to die after this video.
No. What do you think I should say instead?
Fuck you.
It is still going.
Clap again and do it all over. Say something else.
Thank you for watching this video. Again, my name is Dna Heiss. I hope you learned something about DNA replication and PCR, and what interesting concepts they are to the field of genetics. This is the last video in the series, so we've got the whole series of biology stuff complete.
Biology stuff?
Biology stuff. I do suck. It was better than I was. See you later. Okay. So that was DNA replication and PCR. Again, my name is Janae Heiss. I hope you learned something about DNA replication and PCR after watching this video and realized what interesting and exciting concepts they are in the world of genetics. This is the last video in the series, so thank you for watching.